Hello, everyone. Thank you for joining. Good morning, good afternoon, good evening, depends on where you are. My name is Li Man Li. I am the Vice President of Global Virus Network. So on behalf of the GVN team, I would like to thank you for joining us today at our Forefront of Virology COVID-19 webinar series. Before handing it over to GVN Program Director, Dr. Shinhee Kim, to introduce our distinguished speaker, I would like to take the chance and give you a very brief introduction about the Global Virus Network. So the GVN is a non-profit organization which was co-founded by Dr. Bob Gallo, Dr. William Hall, and the late Dr. Reinhard Kurz in 2011 at Baltimore, Maryland. GVN is unique because we are independent and scientific-driven network. Who brings scientists from the academic centers and industrials such as Abbott to work together to advance the knowledge about emerging viral diseases and to develop the vaccines, diagnostics, therapeutics, and so on to protect the public health. So GVN currently is representing 63 centers, 11 affiliates in 35 countries. We meet our mission in 3 ways. By supporting cutting-edge research to train and educate the next generation of the virologists, and to promote and advocate the public health. So to advance our educational and training programs, GVN recently launched a program called GVN Doctors Yanliu and Panzhan Academy. It consists of 5 different programs, including fellowship training, mentorship programs for junior scientists, online short course for emerging leaders, and alumni networking events. So I encourage you to check out more detailed information on our website, and feel free to share with your colleague who may be interested in applying for the program, since we are currently accepting applications. And GVN's mission and efforts also include rapid and efficient response to the current pandemic, and also to prepare for the future ones. So the purpose of these online seminars is to bring our expert virologists, such as Dr. Cloherty, to share their cutting-edge COVID-19 related research. After a 30 minutes presentation, we will have about 20 minutes for question and answers. Dr. Shinhee Kim will help Dr. Gavin Cloherty monitor and select questions to answer. Dr. Cloherty from Abbott is the first director who joined a GVN Corporate Partnership Programs. GVN Corporate Partnership Program is a new initiative that aims to serve as a partnership platform to strengthening the international viral preparedness and also the response between business community, leading medical virologists at the GVN Center of Excellence. So we are the bridge to link the corporate partners and the academic centers worldwide to develop a shared vision and to explore a range of collaboration opportunities. Since the beginning of this pandemic, GVN and Abbott has been working together closely to develop our SARS-CoV-2 biobanking project. Recently, Abbott GVN headquarters and also one of the GVN Center of Excellence, University of Buffalo, are working together to form Emerging Pathogens Discovery Network Working Group. So without further introduction, I'm going to hand it over to Dr. Kim and let her introduce our today's speaker. Gavin, thank you so much for taking the time to participate in this webinar series.
Thank you.
Dr. Kim.
Good morning, everyone. This is my great pleasure to introduce today's speaker, Dr. Gavin Cloherty. Dr. Gavin Cloherty is currently head of infectious disease research at Abbott and a research fellow in Abbott's Volwiler Society. As our Vice President Li said, Abbott is GVN Healthcare and the Pharma Center of Excellence Coalitions. Dr. Cloherty received his bachelor's and doctoral degree in microbiology from the National University of Ireland, Galway. He has worked in Abbott for 20 years by providing scientific leadership in infectious diseases and diagnostics. His research has focused on the development and application of diagnostic tools for HIV and viral hepatitis. His recent research is focused on the early detection of novel pathogens and viral genomic surveillance, or rapid response to future pandemic threats. His research team has developed techniques for the identification and the characterization of unknown pathogens using a megagenomic approach, combining with the next generation sequencing and bioinformatics. His novel approach targets not only to classify pathogens, but also to predict acute and chronic diseases caused by these pathogens. Specific example will be Abbott's identification for novel virus associated with hepatitis viral infection. I also would like to emphasize on his contribution to our GVN's mission and activity. He has been a great supporter for GVN Biobank project. Dr. Cloherty is also leading a new program, GVN Emerging Pathogen Discovery Network Working Group with Dr. Jim Morse, University at Buffalo. This working group is designed to help GVN centers located in low middle income countries to build their research capacity. We will hear more about his exciting discovery soon. So Dr. Cloherty, thank you for giving your talk today. Thank you.
Thank you, Shani. It's very an honor to be here and to have the opportunity to discuss and share some of the exciting work that we're doing at Abbott. So allow me to bring up my slides and make sure you guys can see them in the presenter mode. So just confirm that, okay. So, you know, as I said, it's an honor to be here, and it's one of the high points of my career, in fact, that that Abbott was the first industry partner with the Global Virus Network, and I think we share a lot of the same vision and the same focus for tackling existing viral epidemics and the current pandemic, and also the forward-looking approach of training the next generation of virologists, preparing the way so that we never have to live through another pandemic. I know that's a lofty goal, but that is the aim. So I thought I would give you a brief, for some reason my slides are jumping. I'm sorry. Give me 1 second, please. Okay, so let me do it this way. So I thought I'd start by, can you guys confirm that you're seeing the slide that's titled Meeting the Challenge? Shani?
Yes.
Yes,
Yes.
okay, I apologize. It's showing up slightly differently on my screen, so I apologize for that technical difficulty. So I think I wanted to introduce Abbott ID Core Research to the GVN by describing some of the history, as I said in the title, the legacy of our research. One of the key pillars of that is our global surveillance program, which has been running for over 26 years. So this program has really focused on existing pathogens, HIV and viral hepatitis. And more recently, as everybody, we're looking at SARS-CoV-2 and the variants that are emerging around the world. So the idea is to, and we've done this for many years, is to develop a network of collaborators around the world who can identify samples from infected patients that can then be carefully characterized. The value of this work is to understand how these viruses are changing, how these epidemics are changing, and to apply that from an Abbott perspective to the development of serological and molecular tools. I think this is a very unique program. I don't think there's any other industry program out there that does this on the scale that we do and the way that we do it. We have over 80,000 diverse samples in our biorepository here at Abbott that we've collected in more than 45 countries on 6 continents over the last 26 years. But we're very transparent about the work that we do and collaborative with our partners. All of the data that's generated by the program is shared with our partners. The sequence is uploaded to publicly available databases, and we publish a lot. So As this slide indicates, we've over 125 peer-reviewed publications, and that's actually probably closer to 150 at this stage. So moving to some of the highlights of the last 26 years and focusing more on the recent past, I'd like to draw your attention to a few things. One is that our program was one of only 2 sites that ever discovered and identified and fully characterized HIV group P from a sample in Cameroon. We've added viral hepatitis to the program in 2009 and 2014. And as Shinhee mentioned, we've put our virus discovery program through its paces and identified a novel human virus, HBGV2 or human petitivirus 2. This was found in a woman in Chicago who had sickle cell anemia, had been multiply transfused, had hepatitis C. We identified this virus, but then rapidly developed diagnostic tests and went out to try and understand the significance of this discovery. We searched on all continents, and we actually found evidence of the virus on all continents. We also found for the longest time that this virus was only present, or viremia was only present in people who also had hepatitis C, which was a bit of a head scratcher for a while. We could find evidence that people had been infected with this virus, through antibody testing in other populations, but never a persistent viremia other than in people who had hepatitis C. We discovered that this virus could cause chronic infection by following patients for more than 6 months and finding persistent viremia. But eventually we did find some cases of individuals who had this virus, who were viremic, but who did not have any evidence of hepatitis C. So an interesting case study of the program itself. We did, I think, identify that this virus may not cause any incremental harm to its host. So, but it was a really good proof of principle. In 2015, we also started to adapt the program to do more public health work, leveraging it to better understand the prevalence of viral hepatitis in different countries, working with the Ministry of Health in Georgia on their hepatitis C elimination program. And taking advantage of the biorepository that we have, which I'm really supportive of the GVN developing their own. It's phenomenal to be able to go back over the years and reanalyze samples to see what we found. In Cameroon, we were able to look at samples that have been collected for HIV surveillance in South Cameroon over several years and understand the prevalence of hepatitis C, hepatitis B, and hepatitis Delta, and I'll talk a little bit more about that in later slides. These studies in the Democratic Republic of Congo enabled us to understand the prevalence of hep C, and also our HIV work had some other unexpected findings in terms of the prevalence of potential elite suppressors. So I'll talk about that a little bit later too. So first, the case study of HIV group P. The first case was identified by Francois Simon and as referenced here in the Plantier paper from a 62-year-old Cameroonian woman who was residing in France who had acquired her infection in Cameroon. She was Western blot positive, but there was no detectable viral load. Group O was suspected and ruled out. Sequencing was performed, and then it was identified that this was a case of group P, a gorilla zoonotic transmission, the first that was ever seen of this specific group. We took the lead from the French group and looked in our own freezers and samples we had collected over the years from Cameroon. And in fact, we identified one sample that also had a group P infection. And we were able to get a whole genome, and this was the second sample that had ever been discovered and fully characterized, adding to the body of knowledge. Following on in the Democratic Republic of Congo, we've been doing some work over the last few years, and in that same strain, we actually identified a HIV subtype L, and through the next generation sequencing techniques that we've developed, we're actually able to get a whole genome from this sample. And that enabled the classification, the official classification of subtype L, which was the first one ever classified in 20 years. This got a tremendous amount of media attention. I would say that doesn't appear to have much clinical significance, but it did get a lot of media attention. Similarly, in the same collaboration with the University Protestant de Congo and UMKC with our collaborator Carol MacArthur, we did make an extraordinary observation. In all of the samples that we were getting year over year, we identified quite a high frequency of samples that were antibody positive, but which had no or very little viremia. Initially, we were concerned and thought that maybe we had a specificity issue with serology or a sensitivity issue with the viral load assay. So we did a lot of work to rule out these two possibilities through Western blot testing with other PCR assays, different approaches like this. And these patients were all, our participants were all supposed to be treatment naive, not on therapy. So in the end, we also processed these samples through mass spec to see if there was any drug on board. And we found that actually a lot of these samples we had collected and thought were elite suppressors were in fact on medication, but surprisingly, we found between 2.7 and 4.3, so roughly 3 and 5% of these samples year over year, and these participants had this low level or negative viremia with a positive antibody and no drug on board. So it begs the question, is there something unique about these patients or participants that's enabling them to control the virus? Obviously, we need to do more research here, maybe longitudinal studies, look at the immune system and try and understand this initial observation. Going into our freezers again and our Cameroonian samples, we were able to, if you like, kill two birds with one stone. We looked at the prevalence of hepatitis C year over year, and we also looked for the presence and prevalence of the novel PEGI virus that I discussed earlier. Previous to doing this work, I had been told that the prevalence of HCV viremia was roughly 11 to 15%. This is anecdotally. So we wanted to see for ourselves. And remarkably, we found that the prevalence of HCV RNA positivity in Cameroon, in South Cameroon, was a lot lower. It was more like 4% overall, or 3% overall, 4% in men, 2% in women. So you see immediately that there's a gender bias in terms of HCV viremia in Cameroon. We also were able to identify an age cohort kind of effect, similar to what we've seen in the US, where a lot of the samples that were collected and negative were in the younger age groups, around 20 to 40. And as you get up into the older age groups, 50, 60, 70, you find a higher percentage of HCV positives. And we also, as I mentioned, found PEGI virus in HCV RNA positive samples at a rate of about 15% in this study. The most shocking finding that we had by going back to the freezers was looking at hepatitis delta. So we went back and pulled out roughly 2,000 samples, just a little over 1,900 samples, and that were hepatitis B surface antigen positive, and we screened them for hep delta antibody and RNA. And surprisingly, we found that 46% of those samples were antibody positive for delta, and little under 35% were RNA positive. This was a huge number overall if you extrapolate that out for the entirety of Cameroon. These samples, I must re-emphasize, were collected in South Cameroon, and we saw the same prevalence roughly year over year. We these samples and were able to, I believe if memory serves me, almost triple the number of whole genomes that were available publicly after this study. And what we found was pretty much what we would have expected in terms of genetic diversity and distribution for Cameroon. Obviously, we've now turned our effort and a lot of our focus most recently into SARS-CoV-2 surveillance. As many others are doing in silico sequence analysis of the immense amount of sequence data, yet not enough that's being uploaded to GIS8 and other databases, working with collaborators around the world to identify samples that could be sequenced, adding additional geographies over time and partners. So currently we're working with South Africa and United Kingdom, with group IRSEF in Senegal, groups in Brazil, Colombia. Again, I've indicated a couple of GVN members that are partnering with us in this and other research. India and Georgia has recently been added, and we're continuing to expand our international sites, including some other GVN affiliates in Jamaica and Zimbabwe, working through the paperwork needed to get these sites on board and getting this research up and running. Our sequencing is obviously very active in the US, and we're sourcing samples from most states, highlighting here Illinois, California, Wisconsin, New York, South Carolina, and Florida. We're finding a lot of variants that you have all over the world are now showing up in greater and greater frequency around the US. I would like to highlight some of the learnings that we've taken from HIV and viral hepatitis and we've applied to SARS-CoV-2. So we had developed a method of target enrichment for HIV and hepatitis that enables us to get whole genome or near whole genome coverage even in low titer samples. We've applied this to SARS-CoV-2, and you can see in this slide the benefit of this approach. And the normal metagenomic next-gen sequencing in orange, we really drops off as you get down towards log 5 copies per mil, and even sporadic higher at log 6 and 7. But using this X-gen approach, we're getting whole genomes or near whole genomes enough for classification right the way down to log 3. And we get quite reasonable coverage down at log 2. So this is an approach that we're very happy to have developed for HIV and a learning that we're applying today to SARS-CoV-2. Moving to another pillar of Abbott's infectious disease research and the Pandemic Defense Coalition is hepatitis B biomarkers. And hepatitis elimination in general. This is an area of research that Abbott has been involved in for many, many years, decades in fact, and was the genesis of Abbott's diagnostics division was hepatitis B. But looking at Hep B, there's clearly a shift. There's a lot of focus from the pharmaceutical industry on developing novel therapeutics and even among treaters to try and improve how patients are treated with currently available nucleoside analog therapies. And I put up here the schematic of the life cycle of hepatitis B as an indicator of where we might have some impact with biomarkers and where biomarkers might be useful. So drawing your attention to the bottom here, where you see where nucleoside analog therapies act. They prevent and disrupt the conversion of pre-genomic RNA into DNA, which is obviously critical to make intact virions to go on and reinfect other hepatocytes or infect other people and drive disease. What nucleoside analogs don't do is impact anything upstream. So the transcription of RNA from the covalently closed circular DNA, the production of pre-genomic RNA or its subspecies of other RNAs, the production of other proteins within the hepatocyte, none of that is impacted. So Abbott has taken a very active role in our group in developing novel biomarkers to try and investigate the HBV RNA and also other serological markers such as more sensitive surface antigen detection, first off, and then also isoforms, different versions of surface antigen, and looking at the immune complex of the surface antigen and the antibody to surface antigen. I'll talk first about the RNA assay. So we understood that it was a very needed marker. The existing approach was a manual race-based method, which didn't have a sufficient limit of detection. So we developed an automated approach that works on our M2000 platform that has a limit of detection of 44 units per mil. We standardized it to the WHO standard, and we've been doing a lot of research to try and really understand the clinical utility of this marker in the context of natural history, existing therapy, and future therapies that are in development. So one of the first findings that we had coming out of the gates with this test was looking at when this marker becomes detectable in patients who are newly infected. So these are seroconversion panels, and we tested these seroconversion panels obviously with DNA, but with RNA. And it was interesting to see that that RNA was present pretty much from the get-go. Right from the start of infection, RNA is present in the plasma and detectable, albeit at about 1.5 log to 2 logs lower than DNA. And in an untreated or unresolving case, that as you see in the bottom panel, that trend continues, that the RNA tracks very closely to the DNA, but at about 1.5 to 2 logs lower. And you can see this in the samples in the patients who resolved their infection. Obviously, it also very closely getting a little bit closer to the DNA numbers as you get closer to resolving the infection. So that enabled us to build out the picture and the body of knowledge around the natural history of hepatitis B, and showing that RNA is present from the start of infection right the way through to resolution, and they remain closely in lockstep unless you apply a treatment. So when we add a nucleoside analog treatment, DNA declines very rapidly, but RNA remains pretty stable and then declines more gradually over time, depending on the patient and the therapeutic. So this led us to wonder, what are the potential clinical utilities of a marker that remains detectable after DNA goes negative in people who are being treated with a nucleoside analog approach. So we did this study with King's College in London, and this data has been published. And we found that on the upper left-hand side of this slide, as you can see, when somebody's been on therapy for a long time, their DNA is negative. So we were particularly focusing on stopping therapy. When is it safe to stop therapy? And would somebody have an adverse clinical outcome from stopping therapy, such as a flare? And in this study, there were roughly 20 patients who were on therapy, suppressed for a long time. So DNA was completely negative. Their surface antigen was low or negative, and they were stopped and then followed for a period of time. A subset of them had a severe flare. Almost 20% of these people had a severe flare, which is defined as greater than 10 of the upper limit of normal. Almost 40% had a no to a mild flare, and then about 40% were in the middle. But if you look, DNA gave no indication and no way of stratifying this patient population into who would have a flare and who would not. Interestingly, the only marker that was positive in all of these samples that had a flare was pre-genomic RNA. So the presence of pre-genomic RNA at the time when a physician is thinking about stopping therapy, DNA negative for a period of time, surface antigen negative or near negative, if they're RNA positive, it might cause a physician, give them cause for concern, and maybe rethink whether or not to stop that treatment or be ready for a potential mild or severe flare. So the other question in a similar cohort, again, people who were on nucleoside analog treatment were suppressed for a long period of time, stopping them. Can you predict who would relapse? So in this study, again, of 19 patients who were on long-term therapy, we were able to, only 2 relapsed, but it was interesting that those 2 patients that relapsed also had quantifiable RNA, but greater than 2 logs in this case. Also, they had a low anti-HBs result as well. So the combination of both of those, low anti-HBs and detectable and quantifiable RNA, again, would give a physician pause for concern, and that if I stop this therapy, they're most likely, and in this case, I know it's an N of 2, but they did both relapse in this case. Additional studies looking at entecavir, similar scenario, patients who were suppressed for a long time, and looking at the difference between patients who were RNA undetectable and RNA detectable and quantifiable, and also adding on the layer of surface antigen, quantitating their surface antigen to see if it was above 10 IU or below 10 IU, and looking at that then to risk stratify their risk of reactivation. And in this study, we found that a negative RNA, undetectable RNA, and a surface antigen quantitation below 10 IUs per mil really indicated a very low risk for hepatitis B reactivation, whereas a detectable RNA and a surface antigen or a surface antigen greater than 10 IUs per mil had a high risk for reactivation. So again, similar findings to the study done in King's College London with tenofovir, and then with this study done, memory serves me, in Singapore, looking at entecavir. So moving on to what will these markers not do. So we also did studies looking at hepatitis B RNA and core-associated antigen in treatment-naive patients. Patients in the looking at natural history, they're not on any therapy, and really these markers track so well with DNA that they have no incremental benefit, and no significant value in phases of disease. So really appears that the value of these novel markers come into their own when a patient is put on therapy, either nucleoside analog therapy or some novel therapies that I'll mention in a moment. So I'm going to perhaps gloss over some of these slides in the interest of time, because I want to make sure I allow time for some other discussions. But I'm going to talk about an interesting study we did with Replicor, which is a company that has a nucleic acid polymer therapy, which blocks the release of HBV subviral particles from the hepatocyte. And as we know, the vast majority of hepatitis B surface antigen found in a patient comes from these subviral particles. So we looked at samples from these two small clinical trials to see what we could find out about the HBV RNA and what surface antigen could tell us. And we had developed assays that would detect the small, medium, and large versions of the surface antigen assay. We applied a HBV RNA assay, an ultrasensitive surface antigen assay, and an assay that would detect surface antigen in the presence of antibody complex. And what we found was quite fascinating. First off, RNA, again, provides a good indicator of patients who will remain suppressed once the therapy is removed. So all of these patients were on a nucleoside analog backbone with an immune modulator, and then the application of this NAP therapy. In this patient who was RNA negative, they remained negative when the therapy was removed. It's also important to note that using the surface antigen NEXT assay, which is 5-fold more sensitive than our current assays on market, they were also negative. So it really did a good job of suppressing the surface antigen and the RNA. So viremia did not reoccur. In contrast to this, in a patient that actually failed this therapy following cessation and had a rebound, you can see the RNA was relatively high when the person was on tenofovir and never fully really went away when the nucleoside or the NAP therapy was applied. And this person actually did relapse. So again, it indicates that even with novel therapies, these markers can have value in indicating who's going to be successful when you stop treatment. Interestingly, with these novel markers looking at immune complexes, you can see how the immune complex signal spikes up after therapy was applied as surface antigen is declining, and then it drops down over time and does not rebound as quickly as surface antigen towards the end in this rebound patient. And you can also, if we overlap the anti-HBs to the immune complex, you can see that the antibody to S increases as the surface antigen decreases, and you have this spiking of immune complex that then decreases. You have more of the free antibody to surface antigen detectable. One last point on this study is the ability to look at the different types of surface antigen, whether it be small, medium, and large. The subviral particles would constitute a large number of these small particles. So you can look at the relative abundance of these small, medium, and large and try and understand if it's decreasing or changing over time and what the impact might be coming from. So we can clearly see here that the ratio of small to total decreases, which shows that these nucleic acid polymers are in fact blocking the subviral particles. This is independent kind of evidence that this is occurring in a patient who is successfully being treated with this novel therapeutic, in contrast to a patient who did not respond to the therapy, where the relative ratio remains constant. So I'm going to quickly skip on to the next topic. A third pillar for Abbott infectious disease research is really our Pandemic Defense Coalition that we're standing up, that's evolving and growing out of this research program that's been ongoing for decades. I don't think anybody really needs a reminder of the importance of this as we're living through a pandemic. I think a pandemic that nobody wants to live through again. We have had some, you may call them near misses depending on where you live in the world, with Ebola and Zika. If you're in West Africa, I don't think you'd call it a near miss. And Zika definitely in the Southern Hemisphere, it ravaged through there a couple of years ago. So these outbreaks and pandemics are happening at an increased frequency. There's also an incredible amount of undiagnosed illness going on routinely, as you see in the middle panel, with 70% of encephalitis and 50% of gastroenteritis going undiagnosed. So the program really is focused on looking at new and emerging potential pandemic threats, but also focusing on these undiagnosed illnesses and these chronic illnesses that may have a viral or other pathogen as a trigger. And the approach that Abbott is taking, I think, is unique. And I think this program really is a first of its kind and fits well with the global virus network. We're really focused in identifying these novel pathogens that are viruses and that could potentially be future pandemics, but we're not stamp collecting, as I've heard it referred to. We're not just stopping with the identification, the publishing of a genome or a paper. We're taking it steps further, as I mentioned with the PEGI virus, as we've done in the past. So we follow this cascade, if you like, of identification of a novel human virus or other pathogen, potential pathogen, the rapid development of molecular and serological assays, whether they be rapid diagnostic tests or core lab molecular or serology tests. Abbott has all of these tools at our disposal, so we can rapidly build these prototype tests that we can deploy through our partnerships around the world to understand this discovery better. Where do we see it? If I find it in Brazil, am I seeing it in Thailand? To try and understand, does it cause disease? Is this something we should be worried about? To risk assess it for the public health community and with the public health community, and providing that information to them, as well as the tools to do the work. And if we find that this is a risk and something that we need to raise the red flag on, that we're able to rapidly provide tools to the public health community to try and ring fence an outbreak, to try and act quickly and potentially stop or mute at least a pandemic from occurring and spilling over into the general broader community. So it's a scientific exercise, but it's really a public health partnership. And it's really not intended to be, a pipeline for papers or something like that. It really needs to be action driven, and speed is of the essence. As I talked about previously, our surveillance program had built networks around the world, but primarily focused on existing pathogens, known diagnostics. So collaborators just had to phlebotomize a known positive, provide samples, do some analysis, and we were able to generate very informative data. This pandemic coalition kind of steps things up a notch, because we're looking for new potential pandemic threats, new viruses, new bacteria, new other potential pathogens. We need to partner with infectious disease experts around the world, people who have the skill, who are able to identify a case of interest that needs to be triaged into this program, somebody who's able to identify a trend in a patient population that maybe it's not a very severe illness, but it's worrying, to kind of dig deeper, and then collaborating with a lab that has the highest level of technical expertise that can do the kind of work we're doing here in Abbott Park. And we're dedicated and committed to democratizing all of the protocols and techniques and approaches and resources that we have to sites around the world, and building them up so that we have a very uniform and highly functioning network around the world. And some labs come to and partners come to the table and they're already there. Others need a little bit of helping hand and resource. But in addition to that, as I said, we're democratizing how we do it. So scientists from our partner labs will work with us in our labs, we'll work with them in their labs, sharing all of this information and infrastructure and resources. Which brings me to this slide, which really is an important component of the program. It's the quality. It's the common approach of doing things, so that the data and information coming from one site and one partner is commutable and translatable to another, that everybody trusts the data that's being generated. So, internally, I call it the 3 C's. We had to build the capability of doing the work and the capacity to do it on scale. And then we needed to build the collaborations around the world. So this is what we're endeavoring to do now. We're up and running for about 6 months in getting partners on board. We've been developing these protocols and techniques for many years, which is why we're able to hit the ground running. We want to develop a common case definition, a common way of handling samples, high-level biorepositories, as GVN is helping us with, that the data is processed through very sophisticated bioinformatics pipelines that we've been investing in and developing over the last several years. And that's accessible through the cloud with an interface that our partners can use and take advantage of. It's very good for building phylogenetic trees and publishing papers too, just saying. And then this is the raw material that then will go into help us to rapidly build prototype tests that then can be deployed through the partner network so that we can act in real time. That's actually one of the another driving force for the need to do this work regionally. We can't waste time with samples transporting from A to B with import and export and all the complications that can happen there. Trying to build up that infrastructure around the world is critical. And I think it's very much in line with the GVN's vision and mantra. So the aim of the partnership in this first year is actually quite aggressive and ambitious, but we're well on our way. As I said, we're up and running now for more than 6 months. We've added 9 partners. We've got many more that are in the works, as I say, and expanding our geographic presence every day. So we also want to develop and standardize the methods across these partner network, establishing infrastructure as needed, connecting everybody through the cloud, and then really getting into it and ramping up the active case finding and the work, the analysis, the sequencing, while not forgetting the bread and butter of surveillance of known pathogens. So this program is really a platform for infectious disease research. And while we have critical or core focuses like HIV, hepatitis, and some other pathogens, we're open to working with our partners on what's important regionally and at the country level and even at the institution level. So this can be a very useful platform to amplify virology research. Again, I mentioned building the prototype test. That depends on the other two, on what we find. And I will kind of give you a little teaser that we have found some very interesting, discoveries that we'll be making public very quickly. But the top right-hand corner is really the critical point. It's translating these findings into practice. The publishing is important for transparency, sharing with collaborators, uploading the sequence is important, but translating the practice into findings is really the critical part of this program. So this is a very lofty goal to stop the next pandemic, but I think it's worthwhile. It's definitely worthy of all of our efforts. I don't think anybody wants to experience the pain and suffering and loss that we all have over the last year and are still suffering right now. So I think this is worth the effort. And I really feel that working with the GVN by networking networks, we can get there. By everybody working together and pulling in the same direction is critically important. And I think we've seen what we can do. If SARS has done nothing, it has taught us what we can do quickly and what we can do when we all work together with a sense of urgency. So with that, I think I can stop and take questions.
Thank you for your great talk, Dr. Cloherty. So we already have some questions from audience. The first question will be, would you please tell criteria for architects Abbott test for the detection of a neutralized IgG receptor binding S protein?
Okay, that's a very specific to SARS, I'm assuming. So Abbott has an ACE2 inhibitor assay that we've developed as a research use only tool. if you're asking about the correlation between the quant binding assay and other neutralizing assays, we have data using pseudovirus assays and other methods, print assays, that indicate the correlation is excellent. Really, they track almost perfectly with each other. I don't have that data in this deck at my disposal, but we are actually in the process of trying to publish some of these findings. I'm assuming that was the question.
So have you looked at patient with non-alcoholic steroid hepatitis for your PEGI-virus?
Short answer, no. So this is the question, if I rephrase it, is have we looked in non-alcohol in NASH patients? We have not looked at NASH patients for this PEGI virus, and I think that's actually an interesting idea that's worth following up on.
Next question is that there was a news released from Duke this week that they seems to have developed a vaccine that can protect against many coronaviruses. Are you aware of this update, and can you wait on this news?
So obviously vaccines are not my area of expertise. I welcome the news, and I look for additional data as we all do. It was very encouraging. I think our focus on vaccination is very important, and our focus is on the diagnostic aspect of this and making sure that the variants that may impact vaccine are detectable with our test, and to try and understand the prevalence of those mutants and any others that might show up.
So this is all question we have now. So I would like to ask all the audience, if you have any question, please send us your question. Then we'll forward your question to Dr. Cloherty so he can respond to your question as well. So thank you for taking your busy time and giving us your great talk. And we look forward to your collaboration with our GVN centers.
Appreciate it. Thank you very much. It was.
Yeah, we actually have one more question.
Okay.
The question is, how do you see the GVN working with the coalition?
Yeah, that's just came out.
Actually, that's an excellent question. So already, as you can see from one of my slides, we have several GVN members who are part of this coalition and this effort. And Jean Morse is setting up a working group around this topic. So I think participating in that working group would be a good start. And then we would welcome more GVN members to become part of this work and this coalition, and Abbott can then support the work and help as I just described.
Okay, so there isn't any more questions. So we'll just wrap up your presentation. Thank you.
Thank you, Gavin. Thank you so much. Thank you, everyone.
Thank you. Thanks for your attention. Take care, guys. Stay safe.